Hello everyone, welcome back to the class. The other day we discussed about endoplasmic reticulum, its function. Mainly the function of endoplasmic reticulum is to modify protein and also transport protein from the ribosome or the cytoplasm to the Golgi apparatus for further modification and packaging. So endoplasmic reticulum's first process or first function is to modify protein by adding in precursor oligosaccharide, which was dealt in the last class. Before going into the second function of endoplasmic reticulum, let us brief out what we had learned in the last class. Endoplasmic reticulum, it is the largest single membrane bound intracellular compartment, and it is of extensive network of closed and flattened structure. Enclosed compartment is known to be the lumen, and based on the presence of ribosome, it is classified into rough endoplasmic reticulum, smooth endoplasmic reticulum. Rough endoplasmic reticulum is associated with protein synthesis. Smooth endoplasmic reticulum is associated with lipid synthesis, and smooth endoplasmic reticulum is known to be the transitional endoplasmic reticulum. When cells are disrupted by homogenization, when endoplasmic reticulum is homogenized, then it forms the microsomes. The microsomes which are studded with a ribosome is known to be rough microsomes. The microsomes which are not studded with the ribosome is known to be smooth microsomes. Functions of is for protein synthesis, where the process is known to be co-translational translocation, and there is 5 principal modification of protein: addition and processing of carbohydrates, end-linked glycosylation, formation of disulfide bond, proper folding of proteins, specific proteolytic cleavages, assembly into multimeric proteins. The SER already said it is helping in lipid biosynthesis, detoxification, and calcium regulation. Glycosylation of protein is nothing but attachment of sugar molecule to a nitrogen atom in an amino acid residue of a protein. Consider an amino acid like this, which is having COOH at one side and NH2 on the other side, and it is having asparagine side chain, asparagine ASPL, right? Asparagine side chain, to which the oligosaccharide is attached. Oligosaccharide OS, I'll write it as OS. This oligosaccharide is made up of 3 glucose plus 9 mannose plus 2 naga N-acetyl glucose amine. Just remember this thing. All the precursor oligosaccharide is made up of 3 glucose and 9 mannose, 2 N-acetyl glucosamine. This whole thing is known to be the oligosaccharide precursor or preformed oligosaccharide precursor, to which attaches to the asparagine side chain of a particular protein. I hope this is clear. So end-linked glycosylation is nothing but what? Attachment of sugar molecule. Okay. Next. So this is what I explained now. So biosynthesis of oligosaccharide. Before the attachment of that oligosaccharide, how this oligosaccharide is biosynthesized or synthesized? It begins in the cytosolic phase. You can see this is the cytosolic phase. This is lumen. You imagine this as the is present like this with some attachment. This is lumen, this is cytosol. Now this what biosynthesis of oligosaccharide is happening here. where the it is happening in the cytosol. See, in the cytosol, first what happens is N-acetyl glucosamine, the N-acetyl glucosamine plus 5 mannose is for that N-acetyl glucosamine, 5 mannose is added. See here, the resultant what we are getting is for N-acetyl to N-acetyl glucosamine, 5 mannose residues added, and also it attaches to the dolicol with the help of pyrophosphate bridge. What happens here, the biosynthesis of oligosaccharide begins at the cytosolic phase, and for N-acetyl to N-acetyl glucosamine, 5 mannose is added in the cytosolic phase itself. After that, this unit is attached to the dolicol with the help of pyrophosphate bridge. Okay. Then 7 residue dolicol pyrophosphoryl intermediate flipped to the luminal phase. To explain this, 7 residue can count here. 5 mannose plus 2 N-acetyl glucosamine gives 7 plus dolicol pyrophosphoryl intermediate. It is flipped inside the ER lumen. See, the thing is flipped inside the membrane. Next, remaining 4 mannose and 3 glucose. See here, 4 mannose, 4x mannose and 3x glucose. 4 mannose, 3 glucose are added at the luminal side of the endoplasmic reticulum. Entire glucose 3 mannose 9 N-acetyl glucosamine oligosaccharide is transferred en bloc from the dolicol carrier to an asparagine residue on a nascent polypeptide. Leaving this dolicol as a carrier, this unit will get attached to the asparagine residue with the help asparagine side chain of the amino acid of protein with the help of oligosaccharyl transferase. See here, you can see the attachment with the help of oligosaccharyl transferase. The lipid linked oligosaccharide anchored in the ER membrane will get attached to the asparagine side chain. This is known to be the N-glycosylation. Function of glycosylation. Why this glycosylation happens? In order to fold properly in the ER, it requires some proteins require this N-linked oligosaccharide in order to fold properly. N-linked oligosaccharide confers protease resistance, and also oligosaccharide present on glycoprotein serves as antigens. It is important for cell-cell adhesion. Here also you can see the same process happening. Okay. Next, cell coat or glycocalyx. I have explained this in detail. Next, formation of disulfide bond. There is a disulfide bond formation happening with the help of protein disulfide isomerase, which is occurring in the ER lumen, is also one type of function or modification of protein in ER. Protein folding. It is a third most important modification of protein, which is assisted by chaperone. Prevent segment of a nascent chain from misfolding. Misfolding can cause misfunctioning of proteins. Therefore, in order to prevent them, proper protein folding happens. Important chaperone of HSP70 family located in the ER lumen is BIP binding protein. BIP binds newly synthesized proteins as they are translocated into the ER and maintains them in unfold state. Two other chaperones, calnexin and calreticulin, are carbohydrate binding proteins. They prevent premature and incorrect foldings of segments of a newly made protein. So to understand this folding, normal folding, misfolding, how this chaperone are assisting the protein, we can see or we can elaborately explain using this single image, I think so. See, unfolded protein is here with the attachment of precursor oligosaccharide. First glucose is trimmed off, and second glucose is also trimmed off with the help of glucosidase enzyme. Glucosidase enzyme 1 and 2, and then it folds for after removing of 2 glucose. Then this calnexin will help in binding this, so that it rechecks is it misfolded or folded. The it calnexin will recognize the single glucose molecule of that precursor oligosaccharide. It checks and it removes that glucose unit from it with the help of the same glucosidase. Here N-linked oligosaccharide is present, but there is no glucose. As 3 glucose are sequentially removed, it will again recheck, is it folded correctly or misfolded. If it is normally folded, it sends to the Golgi, where it exits from the ER lumen to the Golgi for further process. If it is not folded properly, then it gets back into the cycle with the help of glucosyl transferase. This glucosyl transferase will add up 1 glucose again, so that it can be recycled, or it can be cycled back for the next rechecking process. So this is to ensure the perfect folding of the protein. Where it is happening majorly in the ER lumen. Calnexin will recognize what? The glucose. This is the major thing. Next, sorry for the interruption. Calnexin and calreticulin bind selectively to certain N-linked oligosaccharide on growing nascent. Recognizes N-linked oligosaccharide that contain a single terminal glucose. The 2 glucoses are removed with the help of ER glucosidase, and the third glucose is removed. And the binding of calnexin and calreticulin to oligosaccharide on incompletely folded polypeptide helps in retaining it in the ER, and the enzyme glucosyl transferase determines completely or incompletely folded state of the protein. If protein is incompletely folded, enzyme transfers a new glucose to N-linked oligosaccharide. The cycle repeats until the protein has folded completely. This is the another image where you can see the cycle also, and the correctly folded protein exiting from the ER lumen to Golgi. This was explained in the last class. Now we'll go in detail about the second major importance of ER endoplasmic reticulum organelle, that is synthesis of membrane protein. The synthesis of membrane lipids happen in smooth endoplasmic reticulum. Most of membrane lipids are amphipathic and they both include phospholipids and cholesterol. Phospholipids are amphipathic molecules, and their synthesis from fatty acyl CoA and glycerol 3-phosphate takes place at the cytosolic leaflet. Make sure you write down this point. It happens in the cytosolic leaflet, and synthesis of membrane lipids happen in the SER. The phospholipids are amphipathic molecule, and what are they are synthesizing? They are synthesizing phospholipids and what? Cholesterol. Fatty acids are first converted into CoA esters. Next, what happens? Fatty acids from fatty acyl CoA are esterified to the phosphorylated glycerol backbone, forming phosphatidic acid, whose 2 long hydrocarbon chains anchor the molecule to the membrane. Next, 2 strategies for attaching head group. That is, first strategy is we can give the example of phosphatidyl inositol synthesis. Second strategy, we can give example of phosphatidyl ethanolamine and phosphatidyl choline synthesis. Phosphatidic acid is activated by condensation of CTP to form CDP diacylglycerol. Activated diacylglycerol unit reacts with the hydroxyl group of an alcohol to form phosphodiester linkage. An activated alcohol is used in the second strategy, phosphorylation and activation of the head group, condensation with diacylglycerol. To understand this, let us move on to the second slide. Here it is. First, what happens? Fatty acids are first converted into CoA esters. So this is the fatty acid binding protein. Sorry, the green one is the fatty acid binding protein. It brings this into the cytosol where fatty acids are kept intact. Fatty acids from the fatty acyl CoA are esterified to the phosphorylated glycerol backbone. Here you can see glycerol 3-phosphate, right? Glycerol 3-phosphate. From that, fatty acids are esterified forming phosphatidic acid. See here the CoA are removed. CoA2 CoAs are removed with the help of acyl transferase, and the glycerol backbone is attached to the fatty acid, which is known to be what? Phosphatidic acid. The molecule is known to be phosphatidic acid. Next, two strategies, as I said in the previous slide. Here the second strategy has been depicted, that is example phosphatidyl choline synthesis is here. Phosphatase, with the help of enzyme phosphatase, this phosphate is removed out. You can see the removal happened here, and there is OH instead of P. Okay, so that is known as diacylglycerol. The phosphate is removed to forming a diacylglycerol. This is what the activated alcohol. Then again phosphorylation happens with the help of what? CDP choline. CDP choline will help phosphorylating it again to form what? Phosphatidyl choline. This is the second strategy of attaching head group. So this is the overall synthesis of membrane lipid. First, what happens? Fatty acids are first converted into CoA esters. It is first converted into CoA esters. Fatty acids from this fatty acyl CoA are esterified to the phosphorylated glycerol backbone. It forms phosphatidic acid, whose two long hydrocarbon chains anchor the molecule to the membrane. Then what happens according to the second strategy? That an activated alcohol is used. How it is formed? With the removal of phosphate group from the phosphatidic acid forms the diacylglycerol with the help of enzyme phosphatases, and the again phosphorylation and activation of head group happens with the help of CDP choline. CDP choline will phosphorylate the thing with along with the choline, which forms the phosphatidyl choline. Phosphatidyl choline is what? It is a membrane lipid. What has happened here? The synthesis of phosphatidyl choline has happened. Where it has happened? It has happened in the cytosol of the endoplasmic reticulum.